Good. Welcome everybody to this PGC Worldwide Lab meeting. I'm happy to present the PGC Working Group today. My name is Barbara Franke, and together with Ben Neal, I co-lead the PGC-ADHD Working Group. We have two speakers today, and I will invite them in a minute. Let me start a little bit by looking back at the field, our ADHD field, and if you look a little bit broader at the child and adolescent psychiatry in terms of genetic work and GWAS work, we have been a little slower than many of the adult disorders. It took 10 years for ADHD to go from the first GWAS that was published in 2008 to the first GWAS that actually had genome-wide significant findings. It was published late 2018, beginning of 2019 in Nature Genetics, and when it was published, it was a real game changer for the field. And since then, the insights into the genetic architecture of the disorder we're working on and its relation to other disorders and traits has increased very rapidly. For me, when looking at this GWAS published in Nature Genetics, one of the most striking findings was the strong overlap, over 90% overlap between the genetics of clinically diagnosed ADHD and the genetics of ADHD symptoms in the general population. For me, it meant that in terms of the common genetic variation in ADHD, we're looking at quantitative differences between patients and healthy individuals and not any qualitative differences. So that means also that studies in the healthy population can be done to understand the genetics and biological underpinnings of ADHD as a clinical disorder. Our first speaker, Tetyana Zayats, will in a minute talk to us about recent work in this direction. Well, the first GWAS to get significant results took us 10 years from starting out on GWAS in ADHD. The second one is going very, very fast. And we've had a data freeze already late last year, and the analyses are currently underway. And our second speaker, Dita de Montes, will give us a little bit of a sneak preview into this second GWAS meta-analysis based on the analysis of part of the findings. So this was my introductory rambling, and I'm very proud now that we have today two excellent female scientists presenting their work on behalf of the PGC-ADHD Working Group. Both are currently climbing the academic ladder very successfully at their own institutes. The first one to present is Tetyana Zayats, who is a researcher at the Analytic and Translational Genetics Unit at MGH and the Stanley Center at Broad Institute. And she's also an associate professor at the University of Oslo in Norway. And I'm very proud that she's also a long-standing and highly valued of the Impact Group, which investigates the ADHD biology across the lifespan. Tetyana's talk is titled Genetics of ADHD Dimensions, Clinical and Biological Implications for ADHD, and I'm reading this from a Word file, and I hope you haven't changed your title. No, you haven't. Okay, so without further ado, Tetyana, go ahead.
Hi, everybody, and thank you, Barbara, for the nice introduction and kind words. And I also would like to thank the organizers for giving me the opportunity to talk here today. I work at MGH in Boston and Broad Institute in Boston, as well as at the University of Oslo. And today I will talk about the genetics of ADHD dimensions. I've divided my talk into 3 sections, and the slides do not move forward.
Could you point, or could you click on your slides? Perhaps then it moves.
Yes,
Yeah.
Sorry. Yes, so I divided my talk into 3 sections. First, I will talk about the ADHD itself, the dimensions, and what was the motivation for this study. And I will then present the actual genome-wide association meta-analysis that we've done. And in the final section, I will talk how we can use these genome-wide data to understand better the ADHD diagnosis, comorbidities, and treatment. So ADHD is one of the most common and heritable childhood onset neurodevelopmental disorders, and it is characterized by 2 main symptoms or dimensions, which is inattention and hyperactivity impulsivity. Because of these 2 dimensions, the clinical representation of ADHD can be quite different across patients. We can see that patients with more inattention or more hyperactivity can react differently to treatment, can develop different comorbidities. They also have different lifetime trajectory of their diseases and different family history. And this gives rise to a dual nature of ADHD. So even though ADHD is considered to be a unitary disease with some subtypes to reflect this inattention hyperactivity within it, we always consider ADHD as a unitary disease, despite this dual nature of it. So when we study ADHD, the first genetic study where we identified the genome-wide significant loci was done in a design of case control, where ADHD cases were all types of ADHD. And we also studied ADHD as a unitary disease when we looked into a continuous phenotype and constructed a score that combined both inattention and hyperactivity impulsivity. When these 2 genetic data were combined and looked into the genetic correlation between continuous and case control dichotomous characterization of ADHD, we saw that the correlation is very high indeed, it's about 94%. And this gave rise to the idea that we could conceptualize ADHD as a threshold disease with an underlying distribution of its symptoms, similarly to, for example, hypertension or hypercholesterolemia. So we would have a distribution of the symptoms with the threshold beyond which we would say that this is ADHD diagnosis. However, because of this dual nature of ADHD, we are not clear about how to handle each dimension in this setup. Some people may have, for example, more inattention and not so much hyperactivity. Then in the distribution of symptoms where they both considered together, the patient may not reach that threshold, but clinically speaking, the patient can be diagnosed with ADHD. So there are still questions about what symptoms and to what extent we should consider to make that diagnosis. What we have also learned from previous studies, genetic studies on ADHD, is that ADHD shows a wide range of correlation with various phenotypes. So it's basically correlates with everything. And this could happen because of these dimensions, that perhaps inattention correlates with certain diseases and hyperactivity correlates with others, and putting them together would widen the range of phenotypes with which ADHD shows high correlation. Alternatively, it is also possible that these dimensions represent perhaps a non-specific component that is present in a variety of psychiatric disorders. So to examine these questions, we have then asked another, and it is about the biology of ADHD dimensions. So what is the biology of these dimensions? And to answer it, we performed a genome-wide meta-analysis of association between the genetics and each of the dimensions. We collected a little bit more than 41,000 children from various data sets, and because of the way how the data is collected in different groups, we have different instruments that measured the dimensions. And I'll talk about it a bit later, how we have taken it into account when designing the study. And I would like here to take a moment to say that this project would not be possible without contribution of many, many people. This is definitely a teamwork, and everything I'll show you after this slide, all our results, is the fruit of many people, work of many people. So we have first constructed a score for each dimension, one for inattention and one for hyperactivity, impulsivity. And then we restricted our analysis to children and adolescents only, to parent-filled questionnaires only, and allowed only one missing item per score. And this was done to minimize heterogeneity of our phenotype. We then examined the association in regression using age, sex, and principal components as covariates. And if there was a group that required additional covariates, then they were added appropriately for each of the groups. So we have taken each of the data sets that we had and first did the genome-wide association study in each of the groups individually. Then, if the instrument that measured the dimension was the same, we combined the summary statistics in inverse variance meta-analysis. If not, we then performed a z-value based meta-analysis and used those summary statistics to estimate heritability and genetic correlations. This is a Manhattan plot of our meta-analysis results for hyperactivity, and below is the one for inattention. We did not observe any genome-wide significant associations, although we did see, for example, here on chromosome 1, that there are some loci that appear to be present in one dimension, but not in the other. And because we didn't observe any strong genome-wide significant signals, instead of speculating of specific loci that may be true and significant for each of the dimensions, I would like to spend the remaining time talking about how we could use the whole genome-wide data that we obtained on the dimensions to better understand ADHD diagnosis, comorbidities, and treatment. So we have asked 3 questions to examine how the genetics of dimensions could be relevant to ADHD. The first one is about ADHD diagnosis. So the ADHD diagnosis clinically is based on the observations of inattention and hyperactivity, impulsivity, but we were curious how much they overlap biologically speaking. Are there any latent factors that could be behind this diagnosis and that are not covered by these 2 dimensions? We have also looked into the genetic correlations between each of the dimensions to see if that could help us understand why ADHD is genetically correlated with so many phenotypes. And finally, we looked into the pharmacogenetics of dimensions in light of ADHD drugs. So to address the first question, we have estimated the heritability of the dimensions, and it is about 5% for inattention and 7% for hyperactivity. We then looked into the genetic correlation between each dimension and ADHD diagnosis, and it is about 95% for hyperactivity and 86% for inattention. We have also observed 73% correlation between the 2 dimensions, which is in line with previous observation from twin studies that is about 80%. Apart from ADHD diagnosis, we also looked at self-report, that people reported in 23andMe whether they ever had ADHD diagnosis or not. And the graph on the right-hand side of the slide shows what is presented in the table just for a better grasping of the observations. What we see is that in 23andMe, it is the inattention that shows higher correlation with ADHD of 75%, while in ADHD diagnosis, it is actually the hyperactivity that is slightly higher correlated with ADHD than inattention. But because we have observed this 73% correlation between the 2 dimensions, when we talk about the correlation of each dimension and ADHD, there is still a contribution of the other dimension in that number. So for example, if we look at this 95% correlation between hyperactivity and ADHD diagnosis, in that 95%, there is still contribution of inattention. So to disentangle these and also And then to get the answer if there are any latent factors behind ADHD diagnosis, we performed a genomic structural equation modeling. We have taken 3 sets of summary statistics, 1 for ADHD diagnosis and 2 for each of the dimensions. We estimated their heritability, and then we looked at the correlation between each dimension and ADHD diagnosis while taking into account the correlation between the 2 dimensions. These are our results, and I will explain these numbers more on the next slide, but for now I would like to highlight this number of 2.5%, which is the unique genetic contribution of ADHD. So if we take the ADHD heritability that is now being estimated to be 23% and normalize it to be 100%, then these 2.5% is the unique genetic contribution of ADHD. 50% are the genetics that overlap between the hyperactivity, inattention, and ADHD diagnosis. 36% is the genetics that overlap between hyperactivity specifically and ADHD diagnosis, and only 11.5% overlaps between inattention specifically and ADHD diagnosis. So overall, we can say that it's quite unlikely that there are any latent factors that would be contributing to ADHD diagnosis after we take into account the dimensions. Our next question was about genetic correlation landscape. And to examine it, we have taken all the phenotypes in LD Hub, it's about 700 phenotypes, and looked at their correlation with each of the dimensions individually. Here I show you just a little graph with some of the phenotypes that we examined to make a point that the patterns of genetic correlations that we observed between the 2 dimensions are not the same. Some phenotypes correlated more with one dimension while others with another. And so we then performed a test to see if there is any statistical difference between these 2 patterns of correlations. And here you can see our results. We did not observe any significant, highly significant difference. And in the table here, you can see the phenotypes where the difference was the biggest, so the smallest p-value that we saw. And so we can highlight some phenotypes like education and intelligence, as well as reproductive phenotypes, autoimmune phenotypes, and obesity. And apart from the LD Hub, we also examined phenotypes that are psychiatric disorders and particularly of interest for ADHD. And to do that, we calculated a polygenic risk score of each dimension in IPSYCH sample and examined ADHD, autism, schizophrenia, major depression disorder, bipolar disorder, and anorexia. In the graph that I'm about to show you, on the x-axis, you will see the z-value of association between the PRS of hyperactivity and each of the disorders. And on the y-axis, you will see the z-value of association between PRS of inattention and each of the disorders. So you can see that the association between ADHD and the polygenic risk of the dimensions is significant, which is what we would expect. But we also saw significant association between PRS of these dimensions and autism and major depressive disorder that are known to be common comorbidities of ADHD. Our last question that we examined was the pharmacogenetics of the dimensions and ADHD, and to do so we have taken all the genes that are believed to be the action points of the currently FDA approved ADHD drugs. In the first column you see the names of each of the genes, and then you see N standing for no and Y standing for yes. So if a drug in a header, for example methylphenidate, acts through a gene, then you will see Y meaning yes, as you would see, for example, for this methylphenidate, it would be the ADRA2A receptor. And if you see N, it means that that drug does not act through that gene. So we have then performed the gene-based analysis in MAGMA. And for hyperactivity impulsivity, the most associated gene was HRT7 receptor that is believed to be the gene through which atomoxetine is acting. However, we did not observe such strong association for this gene in inattention. Instead, the strongest association in inattention was the opiate receptor through which it is believed amphetamine is acting. And when we extend this to the actual ADHD diagnosis, then we see that there is no association whatsoever. So examining the dimensions can give us more detailed information about these genes and perhaps pick up on the signals that we not necessarily can see in more blunt phenotype as ADHD diagnosis combined with all the subtypes together. So in conclusion, we have estimated the heritability of the dimensions that appears to be much less than that of ADHD diagnosis. We have also seen that the dimensions show high genetic correlation with ADHD diagnosis, and there are no latent factors that may contribute to ADHD diagnosis apart from the dimensions. We also presented a landscape of genetic correlations between the dimensions and various phenotypes, with notable differences in autoimmune and reproductive phenotypes, as well as educational attainment and obesity. In pharmacogenetics, if we examine ADHD dimensions, we may see a better granularity in the biological pathways behind the action of the drugs, although it is worth noting that the genetics of a disease can be different to those of its treatment response. And coming back to the conceptualization of ADHD as a unitary disorder with several domains, it may be better to conceptualize ADHD as a spectrum disorder, a spectrum of inattention and hyperactivity, impulsivity with a level of impairment. And this perhaps can already be seen in changes that were done in DSM-5, where ADHD is no longer presented as a disorder with subtypes, but rather disorder with different representations. That could also show that there is acknowledgement of ADHD being sometimes a lifelong disorder and persisting across the lifespan. And with this, I would like to thank everybody who contributed to this work, and I thank you for your attention.
Thank you very much, Tetyana. I would now like to invite people to ask questions, and that can be done through the Q&A chat. You can also, I think, raise your hand, and if everything works fine, Tammy can unmute you. We have one question coming in, and I will read it. How will you explain in the pharmacogenetics that no correlation was found for the DUT gene? In particular, psychostimulants have a huge effect size compared to many other psychiatric disorders, and that's a question. Maybe there are other, may there be other genes related that were not tested here?
So I'm not sure I understood the first part, if it was about a specific gene, but in terms of possibility of having other genes that were not tested here, the answer is absolutely. So we have only tested those genes that are presumed to be the action points of the FDA-approved drugs, but these genes are not necessarily covering all the genes that are behind the treatment response. So this is another point that we have looked at the ADHD and the dimensions that are not necessarily the same phenotypes as the actual treatment response. So we definitely should examine it in a more genome-wide setting, and also in light of the genes that would be associated with treatment response.
Mm-hmm. Would you think that it is surprising that you don't find anything for the dopamine transporter? We've chased the dopamine transporter, I think, for more than 30 years.
For a very long time.
Yeah.
On one hand, it is surprising, but on the other hand, it is also worth noting that these studies are still underpowered, and it is possible that we did not capture all the biology that is there. So I would say it is still possible that we will see that signal if we increase the sample size.
Okay. So there's one question also from Erwin. Will you be extending this study to look at ADHD symptom dimensions in clinically referred samples?
Yes, we will be doing. At the moment, we're working on collecting the sample. So this is a general population where perhaps it is easier to reach a larger sample size. But in clinical samples, we don't always have this detailed information, but we are working on it, yes.
Mm-hmm. Okay. And there's a question from Vera. Do you think that differences in RGS with the diagnosed ADHD and 23andMe ADHD could be driven by age differences. Possibly 23andMe might have more adult ADHDs.
Yes, that is definitely one possibility. And from Vera's work, we know that the people who are diagnosed late in their life, they tend to show different associations with the dimensions that people who were diagnosed early in their life, but it is also could happen because of the high heterogeneity in the phenotype of 23andMe. It perhaps is not as well defined as the clinically diagnosed ADHD.
Another question comes from Brew. What is the current sample size of your analyses? Can you remind us?
Yes, it is about 41,000 children, and we're working on adding about 5,000 more.
Okay, very good. And Katerina is asking, will you also extend to adulthood? So this is, you made a point of using childhood samples, right?
Yes, so when we looked into the population-based samples, the majority of them were children. So at the moment we have about maybe 3,000 adult samples in general population who have information on dimensions. So we are considering using it maybe for some secondary analysis, but similarly to the question about the clinical samples, we just don't have the sample yet, but it is definitely something that we would like to do.
Okay. Another question comes in from Clemens. Have you done sex stratified analyses?
Not yet. So yes, it is something that we will definitely look into as well. And it is something that is planned for the second phase of this project, where we will ask for the analysis in each sex, like females and males separately.
Okay. And another question from Brew. Oh, losing it. Where am I? It came to me as a surprise to see the high genetic correlation between inattention and hyperactivity of more than 90%. I thought it was 70.
Yeah, it is 73%. It was 73%, and in twin studies, and in one small study that was done previously on about 2,000 people, it was about 80%.
Okay. And Katarina is asking when we can read your paper.
Hopefully soon. So I am working on finalizing the analysis of the last 5,000 people, and then it will go out to all the collaborators, and then I can publish it, or we can publish it.
Okay. Another question coming in from Isabel, congratulating you, great work. Does it make sense to test for correlations between each dimension and ADHD diagnosis when the diagnosis is just the sum of the dimensions, presumably with a cutoff? Also, are there actually significant differences in the SNP heritability and genetic correlation? Hard to see without confidence intervals.
So for the first part of the question, on one hand, it's not surprising because, of course, the diagnosis is based on the dimensions. But on the other hand, the point that we wanted to investigate was whether these dimensions and what we capture by the dimensions and what we capture by the diagnosis, do they have the same biology? So often, even within different instruments that intend to measure the same thing, for example, for inattention or hyperactivity, if we take different instruments to measure this same thing, we see differences in the genetics that they capture. For example, in twin studies, the correlation between these data is not 100%. So that was what we wanted to see, and we did see that the dimensions cover, biologically speaking, what we capture in ADHD diagnosis, with very little left for the unique genetic variability. That was reassuring to see.
In terms of that, regarding that question, also I would like to say that in the ADHD diagnosis, in addition to the dimensions, we also have the impairment, right? So whether this is this is part of the dimensions or is actually something that is a little bit different.
Exactly. So that's what we wanted to see, if what the diagnosis captures, because it's a wider picture that the clinician sees, then we wanted to see if, biologically speaking, they capture exact same thing. And then for the second question was about looking into the, I'm not sure I understood the difference between heritability and correlation.
Are there actually significant differences between the SNP heritability and the genetic correlation?
So I'm not sure what is meant here. So,
okay, so perhaps Isabel can say more about that. Well, she may be typing. Can I go to one additional question? Coming from Katarina, you use genomic SEM, but have you considered also looking at phenotypic SEM?
We did not do that, and it is because we are more interested in the biological pathways underlying it. And secondly, we did not do that because we do not have access to such information. So a lot of the groups have difficulties sharing actual individual level data. So a lot of the work was done on just summary statistics, and because of that, the better choice was to utilize it in the genomic SEM.
Isabel can unmute herself.
Yes. Yeah, sorry. So I might have misunderstood about the first part. I thought that the correlation between the ADHD dimensions and the diagnosis, I thought that the diagnosis was the one that's based on the sum score in each sample with just a cutoff, and where there is no.
Yeah, so the ADHD diagnosis was the summary statistics from the study of Dita and Raymond, where it was a case control setup. And
okay, sorry, then it makes total sense.
Yeah,
I take back my question.
Sorry. Yes.
And then the other was just, so when you compare sort of the SNP heritabilities of the two different dimensions or the genetic correlations, I think the plot you showed with 23andMe, it's just hard to see if there really is a statistically significant difference. Have the confidence in errors. I think it's just, we often say this in genetic research, like, look, they're different. But then you look at the standard errors and they're really big, and you're wondering, is that really?
Yes, you're right. I'm sorry, I did not put the actual standard deviations or errors, and I just reported it in a table. We did not see statistical difference between the correlations for ADHD diagnosis and for 23andMe. I just got the results yesterday, so I haven't had time to test it, but it is definitely something that is worth doing.
Okay, great. Thank you.
Thank you.
Okay. We have more questions coming in. Looking at the time, but I think we still can. Tetyana, are you still happy to take questions?
Yeah, of course. I'm very happy that there is interest in this project.
There's one question. Any plan to look at how individual level comorbidity, for example, with major depression, influences heterogeneity in the results?
Yes, this is something that we are also potentially considering to do in the next phase of ADHD GWAS as a diagnosis. So we are collecting the data on comorbidities in all of the samples, and then using these dimensions, we are hoping to see how the correlations behave if we were to take into account the comorbidities and ADHD cases that are clinically diagnosed. So it all will depend on how much data we can collect, but this is definitely of interest and a good idea to examine.
Okay. And one last one. Tinka is asking, saying, thanks for the great talk.
Thank you.
I wondered how unique ADHD is in terms of genetically correlating with everything. Isn't that the case for most phenotypes we study, like intelligence, MDD, schizophrenia?
It is true that we see quite a bit of correlation for many phenotypes, but ADHD still shows much more than what you see for schizophrenia or intelligence. So if you were to look, for example, in UK Biobank at the, let's say, a heat map, where you see the correlations of each of the phenotype in the heat map, ADHD highlights everything, practically everything. It could be, of course, because maybe the weight is, the data has been collected, maybe the way people were scored or diagnosed or self-reported perhaps is slightly different to how people would, let's say, self-report schizophrenia. But ADHD, despite other phenotypes showing a lot of correlations, ADHD still shows more.
Mm-hmm. Yeah, it's a very interesting finding. I think we have to think more about how that works.
Yes, yes, definitely.
Seems to be a risk factor for many things, potentially. Okay, so thank you very much, Tetyana, for this very exciting data. And hopefully, indeed, we will be able to see it in print in the near future.
Thank you so much.
We're going to our next speaker, Ditte de Montes. Ditte is an associate professor at Aarhus University in Amsterdam, in Denmark, where she has worked with psychiatric genetics for at least 10 years. She's also a key researcher in IPSych, known as an interdisciplinary large multi-center research project studying risk factors for the major psychiatric disorders. And we're very happy that Ditte also recently joined the ECNP network on ADHD across the lifespan. We're very proud to have her. Today, she will give us, as I said, a sneak preview of what's coming in the next GWAS meta-analysis, and she will present data on 118,000 cases and 900,000 controls, which is really exciting. So fire away, Ditte.
Thank you very much, Barbara. So as you said, I'm going to talk about new ADHD risk loci identified in a GWAS meta-analysis of 180,000 ADHD cases and 900,000 controls. And I would like to really stress that this is only preliminary results. So first, very shortly about ADHD. As you know, it's a common neurodevelopmental psychiatric disorder affecting around 5% of school-age children. And the twin heritability has been estimated to 0.74. And we have in our previous GWAS meta-analysis of ADHD estimated the amount of risk that can be explained by common genetic variants to 0.22, so really establishing the role of common variants in the risk of ADHD. And also in the previous GWAS meta-analysis of ADHD, this study was based on the large Danish IPSych cohort and 11 cohorts from the Psychiatric Genomics Consortium. So in total, more than 20,000 cases and more than 35,000 controls. And in that study, we were able to identify 12 genome-wide significant loci, the study that Barbara also talked about in her introduction. So in that study, we also replicated and validated our findings by doing a GWAS meta-analysis where we included the data from IPSych and PGC together with samples from 23andMe. And the sample from 23andMe consisted of almost 6,000 cases with self-reported ADHD and more than 70,000 controls. And that meta-analysis identified 10 genome-wide significant loci. 7 of the loci were also genome-wide significant in the primary GWAS of diagnosed ADHD. So what this tells us overall is that for ADHD, we might be close to the inflection point. So the point beyond which there exists a linear relationship between an increase in the sample size and the number of genome-wide significant loci. And it also tells us that we can get extra information about the genetic architecture of ADHD by including other cohorts that has been ascertained in another way. For instance, this self-reported ADHD status by 23andMe, to get extra information and new information about the genetic architecture of ADHD. So the aim of this study is to identify common risk loci and elucidate the polygenic architecture of ADHD based on an updated GWAS meta-analysis of ADHD, including new genotypes from IPSYCH, which is diagnosed ADHD, and from 23andMe, which is this self-reported ADHD status. So IPSYCH, this is short for the Lundbeck Foundation Initiative for Integrative Psychiatric Research. And IPSYCH is a nationwide population-based sample of around 130,000 genotyped individuals. And among these, around 80,000 are genotyped with at least one of 6 major psychiatric disorders, which are autism, ADHD, schizophrenia, bipolar disorder, depression, and anorexia. So we have in IPSYCH practically all cases born in Denmark between 1981 and 2005. And then we also have around 50,000 controls, which are randomly selected from the same cohort. And the cases and controls, they have been identified in the Danish registers, and subsequently we have identified Godfrey cards with blood spots from the study subjects in the Danish Newborn Screening Biobank. And the DNA has been extracted from a little blood spot from these Godfrey cards, and genotyped with a site chip or Illumina's global screening array. And for this study, ADHD cases were diagnosed according to the ICD-10 criteria. So the IPSYCH sample was genotyped in 2 rounds. So the first round, referred to as IPSYCH 1, consists of 14,500 cases and more than 22,000 controls. And those samples were included in the first, or in the latest GWAS meta-analysis of ADHD. And then we have IPSYCH 2, which is the new batch of genotypes, which consists of almost 8,000 ADHD cases and almost 15,000 controls. And here you can see the results from the updated GWAS of IPSYCH 1 and 2. So this consists of more than 22,000 cases and more than 37,000 controls. And in that study, we identified 17 genome-wide significant loci. But also very important, we do find a very high genetic correlation of 0.95 between IPSYCH 1 and 2. So it really supports that there is consistency in the phenotype that is analyzed across IPSYCH 1 and 2, and also that we do not see any technical errors between the 2 batches of genotypes. And then we have the 23andMe data. And the 23andMe data are based on a questionnaire that people that took a test at 23andMe, they answered a question, which was, have you ever been diagnosed by a doctor with any of the following psychiatric conditions? Attention deficit disorder or attention deficit hyperactivity disorder. And the ones that answered yes to that question is a case here, and the one that answered no are controls, and the one that answered I don't know are excluded. So in total, the 23andMe data consists of more than 70,000 cases and more than 850,000 controls. And that GWAS identified 49 genome-wide significant loci. And here you can see the results from the GWAS meta-analysis of IPSYCH 1 and 2 plus 23andMe data. So in total, more than 118,000 cases and almost 900,000 controls. And it was possible for us to identify 85 genome-wide significant loci. And when doing a sign test, we also identified a very high consistency in the direction of association in this new updated meta-analysis compared to the direction of association in the previous GWAS meta-analysis of ADHD. And I would like to highlight just a few of the findings. One of them is the most strongly associated locus on chromosome 3. This locus is located right in the gene CADM2, and also functional annotation in humor also identified several variants that actually were significant, or several of variants located in the risk locus that are significant EQTLs for CADM2 expression in the brain. And CADM2 encodes a synaptic cell adhesion molecule, which is important for synapse organization. So it makes quite good sense considering the phenotype of ADHD. And also when looking into GWAS atlas and doing a PGWAS of the index variant, we could see that this previously has been associated with various phenotypes related to weight and BMI, but also to smoking and cannabis use. Then I would like to highlight the locus on chromosome 7. The locus is located right in FOXP2, and FOXP2 was also identified in the previous GWAS meta-analysis, and this association actually becomes even more strong in this new updated GWAS. And then finally, I would like to talk about two loci on chromosome 18. And two of these loci, when doing functional annotation in humor, the risk loci involve significant EQTLs for the gene DCC and also for MPC1. And as you can see at the left, you can see that one of the loci is located right in the gene DCC. And DCC encodes a receptor that is involved in dopamine axon growth from the striatum to the prefrontal cortex during brain development. So it also makes good sense considering the phenotype of ADHD. And this gene has actually been found to be genome-wide significantly associated in the most recent cross-disorder GWAS meta-analysis performed by the PGC, where the gene demonstrated pleiotropic association to all the 8 psychiatric disorders included. So this suggests that DCC is a risk gene, not only for ADHD, but for maybe for psychiatric disorders in general. And then there is the gene NPC1, which encodes a protein involved in cholesterol trafficking in the central nervous system. And when doing a PGWAS of the index variant in the GWAS atlas, you can see that this variant previously also has been associated with phenotypes related to BMI and weight, and also alcohol intake. Then we also did a metaxcan analysis. So we tested for the association of the genetically regulated gene expression with ADHD. And we tested out, we tested for the expression in 14 brain tissues. So we had 13 models developed on expression and genotype data from GTEx, and one model developed based on data from the Common Mind Consortium regarding expression in the dorsolateral prefrontal cortex. And we were able to identify 84 gene tissue pairs that were significantly expressed in cases compared to controls. And actually the gene that demonstrated the most strongly or the most significant association to ADHD was the gene NPC1, which demonstrated a significant overexpression in the dorsolateral prefrontal cortex in cases compared to controls. And then we also did magma gene set analysis. So we tested out more than 10,000 gene sets. And in the previous GWAS meta-analysis, we did not find any significant gene sets or pathways enriched for ADHD signals. But we were able here to identify 3 gene sets associated with ADHD after correction for multiple testing. And the most significantly associated gene set is a set of genes that seems to be downregulated in postmortem brains from patients with Alzheimer's disease. And then we find a gene set related to lymphoma I don't know really how this could relate to ADHD. And then also a gene set involved in neurogenesis, which makes much more sense considering the phenotype of ADHD, and also that ADHD is a brain disorder. And then here's some estimates of the SNP heritability. So we found a SNP heritability of 0.24 for eyesight 1 and 2. So this is in line with what we found in the previous GWAS meta-analysis. There, the SNP heritability was 0.22. And then we do see a considerably lower SNP heritability for 23andMe of only 0.08. And when estimating the genetic correlation between diagnosed ADHD in eyesight and this self-reported ADHD status in 23andMe, the genetic correlation was 0.64. So a moderate genetic correlation that suggests there is some heterogeneity across these two phenotypes. And in the previous GWAS meta-analysis, we saw the same pattern, this decreased genetic correlation between diagnosed ADHD and self-reported ADHD. And we were able to pinpoint or explain at least a part of this decreased genetic correlation due to different correlations with cognition-related phenotypes. And we redid these genetic correlations and we saw the same pattern. So you can see here that we do find this very significant difference in the genetic correlation with these cognition-related traits. So for diagnosed ADHD, we see a very strong negative genetic correlation of around 0.5 with all these, or at least the three listed here, phenotypes related to cognition. But we also see that for self-reported ADHD, we do see that the genetic correlation is not as, this negative genetic correlation is not as pronounced, and it is actually almost absent when looking at college completion. So what we hypothesized based on this, that some of the heterogeneity might be due to that the ADHD cases that are able to take, or that take such a test at 23andMe is probably a selected group of ADHD cases that are better functioning, so they on average have a better cognition than the average case analyzed in eyesight. And then we also looked at differences in the genetic correlations with aggression-related traits, because this might be related to the ADHD phenotype. So we do see a very high genetic correlation of eyesight with aggression in children and also with antisocial behavior. And this genetic correlation is decreased, it's not as high in the 23andMe individuals. However, the difference in genetic correlation with aggression for diagnosed ADHD and self-reported ADHD is not significant. However, I think this is due to the rather low sample sizes in the GWAS of the aggression-related phenotypes. And when it comes to risk-taking, we do see a difference in the genetic correlation of diagnosed ADHD and self-reported ADHD with risk-taking, where we find a higher genetic correlation with risk-taking in the 23andMe individuals. And it also makes sense considering that risk-taking is actually, or it has a positive genetic correlation with educational attainment. And then finally, also as Barbara said in the introduction, we find here also this very high genetic correlation of diagnosed ADHD with ADHD symptoms in the population. And this is also decreased in the 23andMe sample. And when looking at the genetic correlations in LD Hub, there are no surprises compared to what we found in the previous GWAS meta-analysis. So we do see this very high and positive genetic correlation with smoking-related phenotypes. We do see a very strong genetic correlation with of ADHD with having children at a younger age. Then we see positive genetic correlation with different other psychiatric disorders. And then we have the cognition, where we see this very strong negative genetic correlation. And then the positive genetic correlation with traits or phenotypes related to obesity and weight. And then we also looked at genetic correlations with the UK Biobank phenotypes. And we do find a lot of them that were actually significant. I've just listed a few of them here because I think they're quite interesting. The first group are groups or are phenotypes related to activity, where we see a negative genetic correlation of ADHD with activities such as walking for pleasure and keeping fit and swimming, whereas we do see a positive genetic correlation of ADHD with activities such as restlessness or duration of moderate activity. And how I interpret these results is that we do see a positive genetic correlation of ADHD with kinds of activities that are more random, whereas we do see a negative genetic correlation with activities that involves more planning and commitment and consistency. And then we also, as I said previously, we see this very strong genetic correlation of ADHD with having children at a younger age. And this finding is also supported by some of the genetic correlations in UK Biobank. For example, a genetic correlation of ADHD with being younger when starting to taking birth control pills, and also a positive genetic correlation with having more brothers and sisters and number of pregnancy terminations. So in summary, we identified 85 independent genome-wide significant loci. We identified a new strong risk locus on chromosome 3, as well as interesting loci related to dopamine axon growth and obesity on chromosome 18. And then also previous risk loci on chromosome 1 and 7, the FOXP2 locus and the SOX3, they remain strongly associated. And we found 3 gene sets associated. The top gene set involves genes downregulated in Alzheimer's disease. And then we do see this moderate genetic correlation between eyesight and 23andMe. And we think at least a part of it could be explained by genetic correlation analysis, which implies that 23andMe cases belongs to a selected group of better functioning cases. So lastly, the acknowledgements. A lot of people have contributed to these analyses, a lot of people from eyesight, and of course also thank you to 23andMe and also all the funding agencies. So I think this is all.
Thank you very much, Ditte, for this wonderful outlook on what we can expect from this new GWAS that the PGC Working Group is now doing. 85 loci with only part of the data in, that's really great.
That's amazing.
Yeah. We're running a little bit over, but I hope that people can still stay and ask questions. There are a few coming in. Let's have a look. Somebody is asking here whether the correlation between ADHD and Alzheimer could be due to cholesterol-related genes.
Yeah, it could be. So you mean the gene set regarding Alzheimer's? Yeah, it could be.
Mm-hmm. And the same person is asking also whether it could also be Wnt signaling and which genes were in the list from this cluster. I think that information is publicly available, right? Which genes are in the Alzheimer gene set?
Exactly. Yes. Yeah.
Do you know where people can find that?
I think I saw it in the databases. I can just point you to the slide, so you can see it here.
Yeah.
So you can also see the version of the database that we have used. So it's in there.
Okay, thank you. Thomas Davis is saying, beautiful findings, Ditte. Given the sample size, do you think you'll have enough female cases to do a well-powered sex interaction analysis? And are there any results on X? Oh, sorry, it's Leah Davis.
Yeah, yes. Actually, for 23andMe, I don't have sex-specific GWASs, but I'm sure I can ask them to do that. So currently, I don't have the numbers of females and males that are actually in the 23andMe data. So I don't know if that's biased in the same way as we know it is for the childhood, ADHD, where it's where there are more males than females. Mm-hmm. And we want to look into the X chromosome as well. Definitely.
It will be very interesting, yeah. Katarina says, I missed the genetic correlation with autism. Was it similar as before?
That's actually a good question. And I remember also that we talked about it at the World Congress, because it's not on the slide, it's not significant. I think at least it's, yeah, it doesn't, it's not significant here.
Okay. So what's the estimate?
That's a very good point. I'm sorry, I don't remember on the top of my head.
Okay.
Yeah.
Okay. Have a look. There are more questions. Sarah is saying, lovely talk. Given the very high genetic correlation between college and education attainments, aren't the 23andMe genetic correlation results unusual? Education years has 4 levels. The college phenotype collapses this into 2.
So with respect to, sorry, I'm not sure, is it?
This is about the high genetic correlation with college and education attainments. She's asking whether the 23andMe results are unusual.
Whoops, sorry, yeah.
So you seem to have, for college completion, you have a very low.
Yeah, I think some of this, it's here. I think some of this might also be driven by that the college completion, the sample size is considerably lower than it is for educational attainment. And so that might also influence the results.
Mm-hmm. And then Phil is asking the question, the 23andMe data could also reflect lower accuracy of the diagnosis, leading to weaker heritability and genetic correlation effects, rather than reflecting differences in cognition.
It could also be heterogeneity in the phenotype. I agree on that. Yes.
Sarah, you're trying to communicate. Perhaps can you unmute yourself? Oh, Tammy, can you unmute Sarah? Not possible. Yeah, now you should be able to unmute yourself. Hi, Sarah.
Yeah, so I was just quite struck by, the genetic correlation between the college completion and the educational attainment GWAS is very high, and the results you have in iPsych are very, very similar for those two phenotypes.
Yeah.
But then it does drop a lot for 23andMe. And I do understand that there is a drop in power with the college GWAS, but it's the architecture is still extremely similar. So I was just wondering, I don't see how it could be an ascertainment leading to a drop like that in 23andMe. So I was just thinking, maybe think about some other mechanisms that might be responsible, because it would seem very unusual for that to be driven by ascertainment.
Okay.
Yeah, Sarah, do you have any thoughts about how this could be done? Because I think, yeah.
Sorry, go ahead.
No.
Not right this moment. It's the first time I've seen this. But I will have a think and I will let you know by email if I think of something.
Yeah, because we saw the same pattern in the previous GWAS also, that college completion also had lower RG with 23andMe. So yeah, it seems to be consistent, but of course it was only a very small 23andMe sample at that time.
I mean,
it's
it may be
that,
in part.
Go ahead, Sarah, go ahead.
Sorry. No, it's a genetic correlation. I mean, there is the issue that over 60%, I think it's over 70% of Americans do some degree of college. So whereas in other parts of the world, you have maybe 20 to 30% of people going on to higher education that way. So whether or not that phenotype means quite the same thing in America as it does in other parts of the world is a good question. But this is a genetic correlation rather than a phenotypic or a prediction. So that doesn't really work out. But I will think about it and let you know if I come up with anything.
I would think about it too, yeah.
Yeah.
Howard has a point here. Howard Edenberg has a point here. It says that the 23andMe he thinks is highly biased towards higher SES and education. So may it then be just a informativity thing?
Yeah, that's in line with what we think.
Yeah. Okay.
So you could do an ascertainment correction then if you think it's overly ascertained and sort of correct for the ascertainment compared to the population maybe and see if that comes back up.
And Phil indeed is saying IPSAC reflects clinical cases that have known problems, whereas this is not the case for 23andMe. Could be help to explain this.
Yeah. But that's certainly what it could, and that's also what we actually believe, that I mean that there will be a part of the 23andMe cohort that as I said, they are just better functioning because they're able to, they know about 23andMe and they are also able to go in and take the test, and I think that demonstrates that it's probably people that are interested in genetics or other aspects about you know, ancestry or the other reasons why a person wants to take such a test. So yeah.
Okay, good. So we're 10 minutes over time. I think we had a great session. I think everybody, I want to thank everybody who has attended, has asked questions, and I especially want to thank the speakers again. We don't have possibilities to applaud you, but feel yourself applauded by this group. Thanks again for doing this. And
Thank
have
you.
a great day.
Everybody. Yeah.
Weekend.
Thank you, everybody.
Bye, everybody.
Bye.